University of Georgia. It's my pleasure to introduce this talk by Huang, a graduate student in my lab. We've been working on kind of protein structure and function questions for quite a period of time in the glycoscience space, both in determining empirical structures as well as attempting to model proteins with AlphaFold3 and other tools. So we'd like to discuss our advancements in being able to do AlphaFold3. And so, Chin, you want to introduce yourself? Um,
Um, yeah, sure. Yeah, my name is Ching. I'm a grad student and PhD candidate in a Moremen lab, with Dr. Moremen, of course, as my advisor. And yeah, we have been looking into AlphaFold3 modeling starting roughly last year, late last year when the AlphaFold3 source code had been coming out, and we made some good results and progress, and we'd like to share around with everyone.
Chin, why don't you go to the next slide? So, as everybody in this presentation is undoubtedly familiar, glycan structures have a diverse collection of functions in mammalian systems, starting in the early compartments of the secretory pathway, where they influence folding, solubility, recruitment of chaperones, and quality control, and transport through the secretory pathway. Next slide. They also play an enormous number of roles later in the secretory pathway, targeting to organelles, influencing circulating half-lives of glycoproteins and receptor-mediated uptake and the time of residency at the cell surface, as well as playing roles as ligands and interactions with a variety of pathogens at the cell surface. Next slide. They also play an enormous number of roles in interactions with the extracellular matrix, as well as cell-cell communications. So glycan structures play this diversity of function in biological systems, and it comes about through the large collection of diverse glycan structures. Next slide, Chin. And these glycan structures are displayed in the cell surface and have a variety of different classes linked to asparagine or serine and threonine and other amino acid side chains, and have a diversity of glycan structures that influence all of those biological functions at the cell surface, playing roles in interactions and modulation of a variety of cell functions. Next slide. And in addition, there's a huge diversity of capping structures that play these diverse roles in modulation of these activities. And all of these blue circles are enzymes that are involved in the elaboration of these diverse glycan epitopes and that play these biological roles. But it's important to understand the enzymes and how they create these unique classes of glycan structures. Our lab has been working on these enzymes and structures and functions for several decades now. And next slide. And what we know about the broad classes of glycosyl transferases, they generally fall into these three structural folds called GTA, GTB, and GTC, with a couple of outlier KZ families and GT51, 108, and a couple of others. But the details of substrate interactions and how these glycosyl transferases are acting as templates for glycan structural elaboration is still relatively elusive. Next slide. What we do know is these enzymes create unique templates for glycan synthesis, where they are able to uniquely bind sugar nucleotide or polyisoprenoid lipid substrates and that act as templates and have unique acceptor templates for being able to recognize hydroxyl groups for sugar transfer. And then these active sites have templates for the catalytic mechanism for sugar transfer. And so we have a general understanding of how these reactions occur. Next slide, Chin. But the empirical structures that we have are really only a minor subset of the overall GTs that are available. Only a minor subset of GT structures are known. Very few have intact sugar nucleotides or donors bound, and even fewer have acceptors bound. And very few rare examples actually have trapped Michaelis complexes as a part of the structures. And so there's limited knowledge in terms of the rules of substrate recognition and catalysis that we would like to expand more broadly in understanding how GTs work. Next slide. So what we would like to do and what we'll present today is the question of whether we can leverage AI-based approaches that were largely developed for prediction of protein structures and ask the question whether they are also capable of providing hypotheses and models for glycan structures and in particular glycans interactions with proteins. In particular, we're going to be talking about AlphaFold3 and want to address the question of are there limitations? And as you'll see, the standard syntax for being able to input glycan structures into these programs basically is problematic in its stereochemistry and basically produces structural gibberish. And the question is there a workaround to generate plausible glycan structures? Can we generate models with glycans bound to enzymes or lectins in plausible poses? And how do these models compare with known structures as benchmarks for the validation of the approach? And then what are the capabilities that AlphaFold3 then can provide for hypothesis generation? And where are there caveats, workarounds, and limitations? And as you'll see at the end of the presentation, some things work in terms of modeling, some things don't. Some can be remediated, and some can be worked around in other ways, but some don't work at all. And okay, well, as I was about to say, I was going to hand this over to Chin and discuss our advances in AlphaFold in terms of glycan modeling. So, Chin, why don't you move forward with the talk?
Yeah, let me pull out my laser pointer and yeah, really sorry about the accident happened going on, but yeah, hopefully we still remember Dr. Moremen helped me setting up the stage about modeling glycans and what's the difficulties on the research on the structure and glycan relevant structures. And we talk about that we're leveraging AI-based models and using AlphaFold. So that's where Dr. Moremen introduced and set up the stage, and I'll get into the deep dive of AlphaFold. So AlphaFold really made a huge impact around 2020 as its second generation coming up. It's robust enough that it's generating to a database to provide around 20 million, 200 million around hundreds of millions of structures on the database. And then around that last year, 2024 in May, another impactful paper coming up on Nature that they claim that AlphaFold3 can model arbitrary ligands and modeling all those proteins, small ligands with DNA RNA interactions. But at that time, it's only the AlphaFold3 web server. It's being available at the time. It's more restricted into specific restricted ligands, and not a lot of things can do. It's only until November last year, 2024, that the standalone AlphaFold it's coming up, all the source code available on the GitHub that the users can install it and use the full capacity of AlphaFold. And that's something that we're really interested in and see if it can really model glycans or not, since they say they can model arbitrary ligands. There's several dates I want to mention that in the paper, they said they use a cutoff date around 2021 for training all the models for AlphaFold3 on the PDB database. And between 2021 and 2023, those are the training or validation set it's being used. So in general, I would probably mention referring to 2023 as a cutoff date for the overall AlphaFold3 generation. And this date is important. Those are important dates for us to see what's a validation in our hands that we can use or not, and I'll talk about that later. So what is AlphaFold3 and what's a diffusion model? So the special about diffusion model is in the case of AlphaFold3, it's generating random noises atom first in the space. And then the AI model is a diffusion model that will reverse all the diffusion, denoising it, and then get to our target protein structures. Since it can accommodate atoms at randomly in the space, that small ligands can be modeled as well. So this is the breakthrough of the AlphaFold3. In the original Nature paper, they describe that using this as example, ESTL3 protein homodimer, that the glycosylated N-glycan can be modeled pretty well. Sorry, someone is requesting the remote control. I guess I can just ignore it and keep moving forward. And And then this is let's keep talking about AlphaFold. So yeah, I mentioned that there's unglycosylation being benchmarked and modeled in the original Nature paper. And there's another cases, it's one of like a caveat cases in the AlphaFold saying that the glycans or the small molecule, if there's a chiral center, then the stereochemistry can be flipped and cannot be modeled correctly. So this is something that it's a caveat we to keep in mind, but this is also a sugar that it's being modeled as well. Another good benchmark AlphaFold has been making is that they put out around 30 different benchmarks with bounded glycan protein and benchmark which one is working well or not. Um, it's not clear that what are those models are. It's not clear if it's just unglycosylation as PTM or it's just a single monosaccharides. And since we're more interested into larger glycans, so it's better that we can benchmark something by ourselves. So those are the examples that we have to look into. Those are just portion of them. And we look into the PPB structures. Those are deposited after the cutoff date in January 2023. We model them and compare them with crystal structures or PDB structures available online, but those are not in the training set and align them. And they works astoundingly well that all the ligands, they align pretty well with the crystal structures. And that's give us a good confidence that yes, this tool are working well. However, there's actually a huge gap between starting from looking into the AlphaFold tool and generating those structures. And I'm going to discuss more about those details and how we get here. So in the beginning, I mentioned about AlphaFold3 server earlier, candidate model glycans. So I'm not sure people are familiar with this interface or not. Those are something really accessible online on this AlphaFold3 server that as long as you have a Google account, you can log in and type in your protein sequence and do the modeling. There's only restricted ligands that we can model, but when we click into the protein, select the glycosylation site, we can actually model glycan chain. But when I look into the documentation, something looks really unfamiliar. Something it's that when we look into it, those are not a familiar glycan that we are usually familiar with. For example, this is supposed to be just a beta mannose. There shouldn't be an extra mannose over here, and those should be alpha-3 branches instead of alpha-2. So those are not a really common glycan that we're familiar with. And the bracketing method, it's not something that can really define a glycan linkage. And we found this is a limitation and AlphaFold3 server, although it said it can model glycans, but it just probably cannot do a really good job. So there's one paper published recently that we came across that it's using AlphaFold3 server modeling, trying to model a MEM3 glycan with alpha-3 and alpha-6 branching glycan. But if we look into carefully about our stereochemistry, it's actually the 2-hydroxyl position instead of the 3-hydroxyl position being modeled. And that's not something that that usually existed, and it's not also not the user's intention to do it. So this is one of the limitation that we think it would just be really nice that if AlphaFold3 server can model the glycan linkage correctly, but it's not the case over here. So we look into a standalone AlphaFold, which is a source code available on GitHub that after around half a year after the AlphaFold3 server, they release a source code that we can download it on GitHub and install it on our local machine. However, it's not on every random local machine. It needs to be running on the Linux system. It needs to have really good graphic card, for example, like NVIDIA A100 or H100 to do the run. So it requires some techniques to know how to run the Linux system and get access to those high-end graphic cards. Also, AlphaFold did not release the model parameters. We have to request it, and then they will send it to us. So yes, there are some threshold to model while using a standalone AlphaFold. And after we got everything installed and learn and start looking to documentation, according to this post by Dr. Luca Giovanni, original description on the documentation, they said it's linked to arginine for the N-glycan. That's also not something really common that it exists, but it's not the most common N-glycosylation. It's supposed to be on asparagine for N-glycan that we are more familiar with. And also the documentation about modeling glycans that using standalone AlphaFold or AlphaFold3 server, the syntax is different. So that makes us think, okay, probably that's something that we need to start over, look into it, and see if we can find a good way to model glycans using the standalone AlphaFold. At UGA, we have GACRC, we have good cluster center that Jordan over there helped us a lot on the installation maintenance on AlphaFold 3. And this is a submission file that we use to submit the AlphaFold job. If we look into, there's one line over here. This is the JSON file that it contains all the input parameters that we can use for modeling glycans, including all the protein sequence and ligand that we want to put. All the information will be in there, and AlphaFold will read it and start the inferencing. For those JSON files, there are more documentation over here, and we are focusing on glycans over here. There are ligands, and there are several methods that can be modeled, and we're interested in and see which one will work the best. And those are for general, generic small ligands and not really specific for glycans. So there are some trial and errors that we have to working on and see which one works the best, which one doesn't work. So for standalone AlphaFold, first thing over that we start with smiles. Smiles, it's a single line smile string that it's really easy just after it's being generated, it's easy to copy and paste into the JSON file that we can make AlphaFold to read it and do the modeling. However, when we look into carefully at the stereochemistry being generated by this glycan, this is homosaccharide L-neol-T, which the second residue is supposed to be galactose. But over here, we see this is wrong stereochemistry, that the galactose should be like the fourth residue over here. It's an axial position, but instead, it's being modeled as equatorial, now become a glucose. And that's something we are not interested in too. And this is not the one we intended to model. And we look into binary unglycans. This is something more concerning that the glycans, they are just all various stereochemistry error happen and it's not a regular unglycan anymore. There are all sort of wrong stereochemistry. And we also look into another recent preprint that they use a smile string and AlphaFold to model unglycans. They intended to do hybrid type unglycans, but when we look into the details of, for example, the galactose, the yellow one over here, they all looks really flat. There is no axial at 4 position. And for mannose, we also see something really similar, and we don't think, I mean, at least from those screenshots, we don't think they are mannose. So it is important that to check if the stereochemistry is correct after the modeling and the smiles it just cannot generate a consistent stereochemistry. We're looking into other methods. There's RDK utilities Python script generated by provided by AlphaFold team that can cover smile string insert user CCD, but those didn't work well either. There's another syntax called bonding atom pair. It works surprisingly well compared with the glycan energy minimized structure. Torch angle slight difference, but the stereochemistry, they are all accurate. And those are something that we are interested into, and we want to move forward and see if it's really work well with the context with protein altogether. But first, we want to know what is bonded atom pair. What does this syntax mean? So, for example, if we want to connect or generate the beta mannose linkage over here, then it's the method that connect two atoms together using generated covalent bonding using bonded in the pair syntax. And over here, we want to connect the C1 atom of the beta mannose and the O4 oxygen group with the Glycan residue, and we can generate it, the covalent bonding generated the linkage, and the O1 oxygen will leave automatically. That's a good feature of AlphaFold. And for CCD code, it's a bit more complicated since glycans or monosaccharides are extremely having different conformations. There are different forms as being generated. So each monosaccharide there actually will have eight different CC codes. It would be different alpha and beta conformation, different ring, different pyranols and furanols, or L form and D form. So it's really this is just a simplified table that I pulled out for the um, common monosaccharides from human. And then in the portion of the JSON input file, we can list those CCD code for the glycan that we're interested into, and using providing a connecting records and using bonding the pairs to link them together. So again, the key part over here, it's CCD code, what its chemical component dictionaries. When structural biologists submit their protein structures with ligands to PDB database, it will go through a biocuration process that the biocurators will look into a small ligands if the ligands already exist in database or not. If it's not, then they will create a new CCD entry and then deposit it. And later on, if there is anything similar, other researchers deposit it, and they will reuse that CCD. And so that's how the whole CCD dictionary is being created. And we can search a CCD using this really nice platform, the PDB eChem. We can type in the names or from our small string that we can search it and find the correct CCD code that we wanted to use. In our recent publication, we look into all the CCD monosaccharide in the database, and based on SMFG nomenclature, we put out together a table that when the users are interested in model specific glycans, we can look it up and look it directly. There's other resources online that has comprehensive monosaccharide CCD, including all the modified sugar. Privateer did a good job, and there's an updated by PDB carbohydrate methylation. Those are good resources available online. So as I mentioned earlier, that AlphaFold will automatically leave the leaving atom O1 over here when generate a covalent bonding between glycans. But there is one exception that we found really confusing in the beginning, that when generating silyl linkages using it's an alpha Q6 link example over here, that it will generate duplicated bonds, and that's something a little bit strange. So we are looking into it and see if we can find a way to fix it. And we swap the atom instead of O6 of the galactose, we use C6, and it's even weirder. And the torsion angle, it's just not correct anymore. So we look into it and see if there's any remediation method. In the end, we look into the CCD file. We remove anything that's relevant to O2 oxygen, and then use this as edit form of the CCD, become the user CCD, and then rerun it. And now duplicate bonds disappear, and everything looks pretty good. So now we finally establish the workflow, how to model glycans using AlphaFold3 correctly. Those all the sterility tests about the syntax, we found it many work, and it will be interesting that if those glycans are still behaving well in the context with protein structures altogether. And first of all, we look into the proteins that's in the training set and see if we can replicate the results. And I want to mention that we deposited 47 AlphaFold3 models into a model archive. This is a really good database that the users can look into the models that's being generated computationally. And we also upload all the JSON scripts that the user can download it, run it by themselves on their local machine. And this is good to replicate the results as well. And just want to mention that we have done that. So first benchmark, we are using mannosidases that it's an enzyme that we're targeting the 3' of the N-glycan, and we'll trim off the alpha mannose over here. This is a sterility test we done initially in making sure that the syntax, the script is correct. And then we model in the protein and get the Michaelis structure. Over here, compare with the crystal structure and do the alignment, we can see that the 3' residues, which is residue 3, 4, 6, and 9, and over here that it aligns pretty well with the crystal structures, and that gives really good confidence that yes, AlphaFold can do good job on modeling protein glycan complexes. one thing I want to mention that the 6' we said that it's not interaction with the protein residue. They are more in the peripheral. It doesn't line well. We pull up the B factor map of the protein structures. Crystal structure that the peripheral one, we can see that because it's larger, it means it's more disordered and it's harder to be crystallized well. And for the AlphaFold3 model, we pull out the PODT scoring that it's being less confidence out there. But the 3' having strong interaction with the protein, that's something that's really having high consistency that it works well. And if we look a little bit closer to the residue number 9, the manuals over here, we can see the ring puckering shifted a little bit. And we look into that it's actually making sense that the different ring puckering it's a different transition state of the monocytase mechanism. And there's other studies in other papers that have different co-crystal structures that look into the mechanism using a different homologs of the monocytase. So we think that AlphaFold3 learned from there, and it's applying to the protein ligand glycan construct over here that we're trying to model. So it works out pretty well. Another important thing that we're interested in, it's can we model N-glycans on a glycoprotein, and can we model this glycoprotein into with an enzyme complexes? And the answer is yes. This is something that it's really difficult to get the empirical structure since it's really difficult to generate a homogeneous glycoprotein. So this can be another really valuable thing about AlphaFold3 that we can do the modeling. Another benchmark we want to give it's the MGAT2. This is another glycosyl transferases. The previous one is glycosyl hydrolase, and this one it's glycosyl transferases. That it also the benchmark model aligns pretty well with the protein structures. And if we zoom in closer, it's generating a really consistent, really good Michaelis complex that usually it's really difficult to get its structure since sugar nucleotide as a sugar donor, it's easy to hydrolyze during the crystallization process. Usually it's just being hydrolyzed and cannot be being seen in the crystal structure. So AlphaFold3 really adds a really good value on these models that we can look into the interaction residues. Those are the B factors and PODT map that corresponding to it, that they all looks really nice, align each other. Another benchmark I want to mention, it's ST6L1 as well, that we can generate really consistent Michaelis complexes and other glycosyl transferases that it's also really difficult to work with if it's an innate or intact sugar nucleotide. So now the PDB structures in the training set works pretty well. We can always argue that because it's in the training set. Now, how about the PDB structures? Those are not in the training set. Those are deposited and being released on the PDB after AlphaFold3 paper being published. There are four major classes of different glycan protein interaction protein we're looking into, the GT, GH, lactin, and polysaccharide lyases. We look into those, the criteria we look into in the PDB database. It's that if there is more than 2 residues of glycan, those are not PTM, and we do the AlphaFold3 modeling and align them and see how good they are. So there are several examples that we all publish them in our research publication that I'll just go through them really fast. But if you're really interested in any of the protein, and always feel free to look into our publication. So those are GT examples or glycosyl hydrolase examples that they all can be modeled pretty well. Those are lactins example, and then polysaccharide lyase example. So yes, those benchmarks works really well. That give us more and more confidence that AlphaFold is a really valuable tool to model glycan protein interactions. Another thing that we look into is we have our own unpublished crystal structures in our lab. For example, MGAT1 working by Zhang Ye and Digang in collaboration with Wood Lab that we generated. This is a complicated binary N-glycan MEM3 structure that the 3' over here, it's sitting really close at actually the correct position to the sugar nucleotide, and the 2 position that would be added extended with GlycNet will be facing to a sugar nucleotide. Everything aligns pretty well with crystal structures, and and one thing important is that as we can see that there's no white or gray crystal structure on the sugar nucleotide part. So AlphaFold3 really fills the gap, model the sugar part on the sugar nucleotide, and we can and investigate if the surrounding residues are the catalytic residue or not, and we can look into the catalytic mechanism. We have another crystal structure which is even more complicated. It's a MEM5 structure that AlphaFold3 model also aligns pretty well with crystal structures that they just works well. And another really complicated example unpublished structure working by Stephen in Houghton Wenger lab and collaboration with Food Lab altogether, that it's modeling the B3-glycosyl transferases. It's a unique enzyme that model on fucosylated TSR domain of the protein. And again, the sugar part on the sugar nucleotide, it's not being visualized, or it's not showing in the crystal structures. And it's really nice that AlphaFold3 can fill the gap and then provide more valuable insights in the structures. So other than that, we have other ongoing projects in our lab that we use AlphaFold3 models and corroborate our empirical experiments, and we can design more experiments. For example, this is a CH3S1, CH3Y3. It's a contracting sulfate extending polymerization enzyme that it's forming a heterodimer. We have cryo-EM structures, but it's really difficult to get the ligand bound in there and get the clear electrodensity. So AlphaFold3 models really fill the gap that we can look into the mechanism of the binding mode and the catalytic mechanism more in detail. And based on those models, we can generate hypotheses and design downstream experiments. The same thing toward GALT1 and GALT7. This is again another heterodimer for homoglycerol extension. And we're also working on other GT31 enzymes that it has various sugar nucleotide donors, various glycan as acceptor, and this is something that we're really interested in looking into. And we all got really robust results of AlphaFold3 Michaelis complex models, and for us to design downstream experiments and validate if those models are valid or not. So I hope all those examples kind of like give the audience and give the users that yes, AlphaFold3 is a really powerful tool that when it works really well. So next thing that we're interested in looking into is other than the glycan classes I just mentioned earlier, can other class of glycans being modeled as well? So first thing we look into is glycolipids. Either it's linked with ceramide, if it's glycosphingolipid, if it's with phosphate, chitobiols, or GPI interprotein, all of them can be modeled pretty well with correct stereochemistry. And the examples over here is the lactin that's recognizing the global edge glycolipid and aligns compared with the crystal structure. It modeled pretty well. Another example is LH1. This is mannose transferases that acting on chitobiols with phosphate on the reducing end. And this doesn't have a crystal structure in the PDB database, but the Michaelis complex forming over here looks really consistent. And yes, we think AlphaFold3 can model glycolipids pretty well. Another thing we look into is O-glycan, either O-GalNAc glycans, core 1 to core 4, or extended O-glycan, or the glycan with xylose modification, the HSCS linker, or with O-mannosylation, and all the glycosterochemistry, they all being modeled pretty well, and it, it, it demonstrated that AlphaFold can model on O-glycans as well. Another example that we look into and interesting in it's investigating the cis masking and trans activating activity of SELECT2. And the example over here, it's that after having a 6-O sulfation adding into the system, that the sulfate over here, it's interacting with the lysine. At least they are in a really close distance with the lysine over here that is not being shown before. And this can be explained why it has a stronger activity or binding affinity compared with the one without sulfate. And of course, this is just a model. It needs more empirical experiment design, but this is something that gives us confidence that yes, AlphaFold3 can be good at generating structural hypothesis. So another thing that I want to jump into is that yes, using the bounded interpair syntax can model glycan pretty well. However, it can start to be really complicated that when there are more glycans in the system, there are more N-glycans on the glycoprotein, and generating those by just typing them one by one can be error-prone and tedious. So we generated a web tool that basically we can pull in the protein sequence and then pull in the glycan sequence that either it's a pre-built template or using sugar jar to try and export it directly. It will generate it automatically compiled the connecting record and the JSON file directly in our web tool, and we can download it and send it to AlphaFold3 to do the direct modeling. And we submit and we publish, we deposit it to a bioarchive and everything. It's already online, and you can scan the QR code to use the tool directly, and everything it's on the GitHub that you can also download the building by yourself. I don't think I have much time going through all the details. I'm just going through, I guess, two of them. That the examples over here is we can add the protein tab using UniProt sequence ID to pull in the protein sequence and detect the sequence, N-glycosylation sequence, and then pull in the pre-built templates that we can generate it. In the case over here, it's a 2,6-link sialic acid binding N-glycan that, as I mentioned earlier, that the CCD needs to be fixed a little bit, and then everything can be automatically compiled all together just with several clicks, and we can download it and upload it to AlphaFold. So another thing is that yes, not just pre-built template, we can draw glycan using sugar jar as well. And then for all glycosylation sites, since it doesn't really have a robust and well-known sequence like N-glycan, we can manually add it and then typing the protein amino acid sequence, and then we can using again sugar jar to build the N-glycan. And there is pre-built all glycan templates in there. We can import it directly, and then we can export the JSON file to AlphaFold and then do the inferencing modeling. It's the same thing for adding the ligands. I'll just skip it. And another thing I want to emphasize is that we can draw the glycans, but if we want to validate it, if that glycan is existed in the database or not, we have a lookup function over here that we will look up the glycosity data being exported, generated by sugar jar, look into either glytucan or glygen database, and it will automatically generate the ID for those two database and also the web link. And this is good for documentation, and it's good for reproducibility for future rounds as well. And another thing I want to mention is that Ligand has a protein glycoform database that if we are not clear if, for example, a glycoprotein, which N-glycosylation site is being modeled, and what glycan is being added on each glycoside, and this is a really valuable tool that we can look it up and then and know which glycoside, which glycan we should model. So just want to credit that. Now I'll go into the caveats part. Since AlphaFold3, everything looks really good, really nice, but what are the caveats and what are the limitations? So first of all, yes, modeling might not be working. In the AlphaFold GitHub, there's several discussion also in the original paper that I mentioned that if it doesn't work, it's worth trying more seed that Brown generating more models, and the internal AlphaFold3 ranking will generate a better model, higher scoring model, and sometimes it can solve the problem. And in our JAAG tool, we have a function that over here we can easily generate either sequential seeds number or random seeds number that it will help us streamline the modeling process. The second thing is that the stereochemistry and chirality issues that I mentioned earlier, that there is no really a scoring methods that can flag the errors, the PODDT, the PAE, IPTM, or PTM that I look into. None of them, if the stereochemistry is wrong, that those models, those scoring cannot really reflect it um, um, if if it's generating a if the stereochemistry it's wrong. For example, if galactose being modeled as glucose, it cannot be flagged. So we have to be really careful when assessing our models, when inspecting it. We need to be really cautious that if the stereochemistry is still correct. Another thing is that the ring conformation, torsion angles, sometimes it can be a little bit off, and there's a good, thankfully there's a good tool by Privateer web tool. Basically, just upload our model and we generated a report that if the linkages, torsion angles, it's sitting in the common area or not, and this is a good way we can assess if the model is good or not. Another thing I want to mention is that if we are modeling an glycoprotein but without keeping any unglycans on it, and if we're model interacting glycan as a ligand, which happens to have a GLINAC as the reducing end, most of the time that glycan will not be sitting in the active site, but instead it will become one of the unglycation site. So it's more advisable that when modeling a glycoprotein, it's better to put all the unglycans on the unglycation site. So we can focusing on the modeling of glycan protein ligand interaction instead of generating some other artifacts. Another thing is that AlphaFold3, it's really powerful. But it's mainly data driven from the training set. It's being generated, being trained, and there's not really a physical chemical properties are being considered. There's a recent Nature Communication paper published that they look into the active site with a ligand dunking there, and they remove all the interaction residues, or they add bulky residues to all the interaction residues, or change their properties. We can still see that the green one is an AlphaFold3 model, the gray one is a crystal structure set. The green one is still sitting there, and even though the physical chemical properties is completely changes in the active site, they're just not looking to one protein, two protein, looking to hundreds of protein that can see the pattern. So yeah, this is one of the caveats that we need to keep in mind that yes, we need to analyze those models with cautious that if they are really forming a meaningful interaction or not. Another thing is that it's for AlphaFold3 cannot generate the combining affinity, and probably most people have known that those two can generate the KD matrix, how good the ligand's binding to the protein structure. However, there's no equivalent bonding interpair methods in both two that when I link all the unglycans or all the monosaccharides all together as main 3 glycan, we can see that there is all the oxygen, there's still being remained over there and it's not leaving. Compared with AlphaFold3 models and the crystal structures, we can see that the yellow trace it's opposed to modeling results that it's not aligning pretty well with the crystal structures, and probably it's because of the living atom. It's not the function is not available yet for us to model glycans correctly. And in the end, I want to mention that yes, AlphaFold it's a black box, but it requires empirical validation to validate if the model is correct or not. But it's a really highly valuable tool that can generate structures for us to generate some hypothesis and to see if the structure catalytic mechanism is correct, the binding pose is correct or not. It's really a bridge to testing for us to generate a hypothesis, and we can use those models to validate the downstream experiments and to look into other bigger pictures of how this protein is really working and how we can engineer it, or and all kind of applications going on. Again, I want to mention that yes, we look into all the benchmarking marks that not in the training set, and they all aligns pretty well to the crystal structures, and AlphaFold it's powerful as long as we treat the models with caution. In the end, I want to thank everyone contributing to the project. And thank you for the funding source, and thank you for Glycan for the invitation, for having us to giving a talk. And sorry about